Now it's recording. Okay, and what gives it away is this little red dot here. They could make it smaller, you know, they could make it microscopic. It's already a million, but that little red dot pulsing says that it's recording. All right, so welcome again. And always like to begin with a little prayer. Come, Holy Spirit, fill the hearts of your faithful and enkindle in us the fire of your divine love. Amen. All right, so this is the Bio 510, which is a bioethics course in the master's program. And I was going around already. Elisa and Kareem and Luis have told us their goals in finishing. Maybe would you like to tell us a little bit?
Well, I have a degree in neuroscience, and my goal ultimately goes to become a neurologist, of course, and go to medical school.
Okay. So you have a bachelor's so far or master?
Bachelor's and getting my master's done now.
Right.
Yes.
And the master's, this one.
Yes.
Okay. So you're also aiming for medical school.
Yes.
All right. Good. So many of the topics that we're going to cover in this course are relevant to the medical field and healthcare, all right? But also more broadly, this issue of bioethics. Okay, before I get into the subject matter, which I love, let me give you a little bit of background, just a synopsis, because everyone has an opinion, and we respect all opinions, but not all opinions are informed. All right, and so when we talk about issues in bioethics, which typically involve at least the health of someone, if not the very life of people, then it's important to have competence in speaking with these issues. All right, and so my background academically, very briefly, I am a Catholic priest in the Archdiocese of Miami for 36 years, and I have 2 terminal degrees. I have a degree in moral theology from a university in Italy. It's the Jesuit University in Rome, Italy. And then I also have a doctorate in genetics from Purdue. Because I have a background in biology before the priesthood, before going into the priesthood, I studied biology, MDC, Miami-Dade College, and then FIU when it started in 72. Remember the bumper stickers, FIU in 72. I had it on my 1960 Ford Falcon. That cost me $100. And back then, I just wanted to be a marine biologist. I don't know if you've ever heard of Jacques Cousteau.
Yeah.
That was my hero, okay? What I wanted to do was scuba dive in the morning, sail in the afternoon, and party at night. That was the plan. So I got all the way through with a bachelor's in biology back then, and was teaching high school biology and so forth, and then eventually discerned a vocation, going into the priesthood. At that point, I said, well, biology is going to become a hobby for me, because what does a priest do with biology, right? Then what happened is, over the years, bioethical issues started coming up. You may have heard of Dolly, not the actress, but the sheep. Was the first cloned mammal, right? Dolly the sheep was the first cloned mammal at the Roslin Institute up in Scotland. And so I said, well, sheep is a mammal and we're mammals. So biologically, to clone a mammal in the lab is the same exact procedure for a human or for a sheep. So should a human be cloned? And that's where bioethical issues started coming up. And so the long and the short is that I asked my bishop if I could go for further studies twice, see if I could learn it eventually. The one time I went to Rome to get a theological degree, and the second time to Purdue for a scientific degree. So at that point, my degree in Purdue was, the second one was in the early 2000s, 2001 to 2005. So imagine in 30 years, my bachelor's in biology was from the 70s. In 30 years, a lot had happened in biology. Well, the first thing I had to learn at Purdue was actually how to pipette, because we didn't have pipette. We had glass capillaries back in the 70s. There were no pipettes. So I had to learn how to pipette. That was the first thing.
I heard sometimes people, with like actually suck.
Yeah.
And they had to be very careful. of
Very,
course.
very. And they had a little line where the volume was, and there were little bulbs that we would use. But sometimes in desperation, people would suck, and it's bad news. Thank God it was typically microns, so it wasn't that bad. But yes, there's a lot of catching up that happened, but I love that experience. It was just spectacular to come up to date in a field that is very relevant and expanding. It has a lot of ethical issues, as you can imagine, in genetics. Because what I say is, what used to be science fiction a few years ago is now a reality in the lab. So it can be done, but the bioethical question forever is going to be, should it be done? Should it be done? Let me stop for a moment. Elisa, sorry, is it Elisa or Elisa?
It's Alyssa.
Elisa. Okay, so Elisa, can you hear me all right and follow?
Yes.
All right. If at any point you can't hear me, just yell out something, all right? Or send a text to Luis or something, because sometimes I tend to talk fast or not look at the microphone, wherever that microphone is.
Okay.
All right. Yes, so bioethics. Let's talk a little bit about bioethics. Bioethics, as you can see, it's a compound word, right? And these compound words typically are complicated. There's biology in it, right? So there's a biological component, but then there's an ethical component. So biology is one of the empirical sciences, meaning that it can be measured. And that's the realm of empirical sciences, that anything that can be measured, weighed, etc. That's the realm of empirical sciences. And with regards to life, but speaking of organic life, not the life of the angels or something like that, this is organic life, right? That's biology, the study of organic life. Ethics is one of the 5 branches of philosophy, together with ontology and metaphysics and logic and aesthetics. So ethics and empirical science and philosophy in principle don't have anything to do with each other. But now when we put them together, it's an emerging field. It's a new field that is thoroughly interdisciplinary, right? So one needs that dual competence, the competence in the empirical sciences and the competence in philosophy. By the way, so my first doctorate is in theology, and yet to do theology, we need philosophy, all right? So anyone who with a theological degree implies that we have a philosophical background. In fact, that's what the PhD means, right? It's a doctorate in philosophy. So we need to philosophize a little bit or think through the issues. It's another way of saying that we need to analyze these issues. Again, going back to the can and may, what I say is that the distinction between can and may is not just a grammatical distinction. We can do many things in the lab today or in the field, but should we do them? May we do it? That's the key bioethical question, right? Should we do them? And then depending on our philosophical system that we're going to use, either implicit or explicit, the answer will be yes, no, maybe, depending on certain circumstances, etc. So we need to engage in the ethical analysis. And the challenge is that ethical analysis takes some time, yet the biology of it and the technology of it is moving forward relentless, and technology doesn't wait for the ethical analysis necessarily. Sometimes something goes out already into the public, like for example, in vitro fertilization, which is heavily technological, right? And we'll be discussing that, God willing, next week. And before we know it, it's out there already and people are using it, but should it be done or not? Now, so we're typically in bioethics, we're playing catch up after, like they say, the genie is already out of the bag. How do you put it back in? Hello, good morning, welcome, and see, yep. All right, so this is the challenge in bioethics, all right? To try to stay up to date with this field that is moving very quickly. Because when you think about it, of course, a lot of what's pushing it is technology. For example, I don't know if you saw the news just last week. A couple of days ago, the Chinese landed their own rover on Mars. We have a number of rovers over there, a number of jeeps that are going around taking photographs and sending them back to Earth. And now they landed one there too. And so that's a challenge, right? That's a challenge. And so these are the challenges that we need to face and do an analysis and see whether in the end this technology should be applied to the human or not. For example, you've heard of CRISPR, I'm sure, right? Should we do CRISPR on the human genome? Any of you hear about Lulu and Nana? Anyone hear about Lulu and Nana? The twins that were claimed to have been edited from China a couple of years ago,
We
right?
talked about it. It was one of the last subjects in the prior class.
Oh, yes. What was that prior class?
It was molecular. Oh, cell bio? I don't
Okay.
know.
And who was teaching that?
Dr. Mall.
Dr. Mall? Okay. Did she get into ethical issues or was strictly staying on the science of it? Strictly science, right? Okay.
But we know that he got like arrested.
Yeah, right. He did. Okay, well, we'll be touching on that a little later on, basically. But these are the challenges. I just pose it as one of the challenges. We have the technology today to do incredible things, right? Should we or should we not? Should we do it on animals and plants? Should we do it on humans, etc.? So that's a little taste of bioethics. We're going to be a little fast-paced. I'm going to assume a lot about your biological background so that we don't spend too much time there. Okay. Normally, I also have a master's in, teach a master's in bioethics here. We have a program just like this one. We have a master's in bioethics where we go into greater depth in all these issues, but the students there have a broader background. Some don't have any biological background after high school biology, sophomore biology. And so I need to do more extensive biology there. Others do have the empirical background, but not the philosophical, theological. So
Is that a thesis program?
it's a non-thesis. No, it's not a thesis. It's 30 credits, and I can tell you more about it if you're interested later. I'll give you some information on it. Okay, how many credits is this program? 30 or 30? Okay, so that's the minimum for a master's, and we try to keep it like that because you people are paying per credit. So that's it's a market thing. Okay, let's move on to any questions or comments. If you don't stop me and jump in, I'll keep going. All right, that's my style. I also wanted to talk some technicalities about the schedule without having to spend too much time on this. First of all, an hour, what is this, an hour and 15 minutes. It's really, really short if we're going to keep it to 8 weeks. And so I'd like to carve out another 15 minutes, more or less. And I think we could do that if we go, for example, if we stay with the current schedule, instead of 12:05 to 12:15, instead of starting at 10:50, 10:45. So maybe 5 minutes earlier, 10 minutes later would give us an hour and a half. All right, that's one thing. The other thing would be to move the days around because I also have the Summer Research Institute going on, which is on hands working with some undergrads in our forest. We have a unique /pine forest here that we're trying to preserve. Luis knows a lot about it. And it's super invaded with foreign species that we need to clear out by hand. So it's labor intensive. And the morning is the best time in general to work in the forest in the summer because there's generally less rain, a little less hot and so forth. But these 2 days, Monday and Wednesday, having to stop around 10 a.m., a little after 10 for the class is a big burden for me and the 5 students I'll have there. So I wanted to ask right away if we can work out an alternative schedule. For example, can we move it later in the day, in the afternoons, for anyone?
same days?
We can try to stick with the same day. Another alternative is I teach on Saturdays, the master's program in bioethics, Saturday morning. And if people are open to early Saturday afternoon for one of the sessions, I don't know, like from 1 to 2:30 or something like that. You can bring your lunch and eat if you want. Or is that going to be too, getting into the weekend, too invasive? You tell me, you prefer to keep it Monday through Friday sometime? Prefer and have the weekend for yourselves and for other commitments? Okay, so let's discard the Saturday then, Monday through Friday. Can we move to the afternoon, to the early afternoon? Or is that too cumbersome? No? If you're night owls and prefer to sleep.
All our classes up to this point have been afternoon.
They have been,
Yeah.
really? Okay, so you're kind of used to that. All right, do you have any other course? You have one more, right?
Yeah,
Field
but I
research.
think Dr. Plunkett doesn't start till the 28th. Or at the first week of June.
28th?
And that's our own schedule.
Oh, because it's field, right? It's field experience.
We're supposed to have a research with Dr. Mull or something like that.
Well, those are workshops.
Yeah.
That will
Workshops?
alert you ahead of time.
I see, so she can work around our schedule, hopefully. Right, okay. Well, so we can explore the same day, but in the afternoon, Monday and Friday. All right, also saying this, you notice I record every session and then I download it and make a YouTube out of it and send you the link automatically. So that if you miss a session, you can always see it afterwards. But I highly discourage that because I want you to participate actively. Even if you have to participate by Zoom, like Alisa, right? Then you can do that. The ideal to me is face-to-face, directly in person, but the second ideal would be through Zoom, live through Zoom. And the third is you watch the video. Which, by the way, as you're thinking of the schedule, I want to say also the assessment for the course is basically I ask for a summary of each lecture, all right, a summary, 1, 2, 3 pages in a Word document. You send it to me, download it and print it and correct it and send it back to you. And then that's going to be on each summary will be graded on a 100% basis. And the final grade is just the average of those. So there's no paper, there's no presentation, anything else like that, just that summary. And what the summary does, it gives me an idea of where you're at, each one. And if everybody's missing the same point, that's on me because I didn't explain it well enough. So it's feedback for you and feedback for me. You okay with that? Yes? All right. Easier than having to do a paper, another paper, etc., or a presentation, just summaries of every lecture. All right. And you have the PowerPoint. I give you the PowerPoints also. That's another thing. I'm kind of a little all over the place. But the idea is this. I want to hand on the competence. Okay. I don't want to hog this competence. I will be generous, not stingy. And the more people know about bioethics, the better it is, because sooner or later you're going to encounter these issues. And so I want you to become experts in it. All right. You're now newbies and so forth, but I want you to become experts. And that's why I give you the PowerPoint so you can use them later to give presentations or to talk to people formally or informally. It's up to you. But they're yours to keep and edit and augment as we go along. As the issues continue to rise, you can make folders and whatnot and keep adding to that PowerPoint, whether it's beginning of life, end of life, healthcare, and so forth. So you can use later on for yourself, even settings like family and friends, they're going to ask you, beginning of life, most of the issues are now end of life coming in, questions, should we disconnect, should we not disconnect, and so forth. Okay, all right. Okay, so back to the schedule then. How about we stay Monday and Friday afternoon? What's good? 1, 1:30, 2 to start? Thinking that we're going to go for about an hour and a half.
Whatever they decide is fine.
You're open? Everybody's pretty open. Okay, Lisa, you have any particular preferences?
No, just the afternoon's okay with me.
Afternoon, all right. Okay, keeping in mind that generally it tends to rain more in the afternoons on in the summertime. So I can use the morning to work with students in the forest, with the undergrads. And by the way, you're also welcome if you want to help out in the forest and do some volunteer work. You can log it as service hours if you do service hours for anything. All right, but you're welcome.
Expect me popping in and out.
Yeah,
'Cause
absolutely.
I'm
Yeah,
in
it's
USRI.
great. We have a unique setting here with a slash pine forest. Slash pine is the only conifer that has survived and thrived in South Florida. And they go back to the recession of the last ice age, 10 to 12,000 years ago. So it's quite a privilege to have these majestic trees here. Many of them are what we call specimens because of their size. One person is not sufficient to go around that trunk. At least two people, some of them will take three people to go around those trunks. They're just majestic creatures, and we have about 15 acres of them here. We're trying to preserve long term, little by little, but we're working on it. Okay.
And so the, I'm sorry, just for the assessment again,
Mhm.
it's due the following class, so Monday lectures due Friday.
Right, let's see how we work it out so that if I give you, I need some time to correct it. For Monday to Wednesday midnight, is that too short? Let's say for the Monday lecture, Monday afternoon, after the lecture, you'll have the PowerPoint, you have the PowerPoints beforehand, you'll have the actual lecture available. So Monday and Tuesday and Wednesday, that will give you essentially 2.5 days to do the summary. Would it be due by Wednesday midnight, for example? Wednesday midnight, the Monday lecture? And then, so that gives me Thursday for correcting it and having it back to you for Friday, right? And then the Friday one would be Saturday, Sunday. Okay, I need to correct it. Sunday morning, so I can correct it sometime on Sunday. That gives you Friday afternoon, Friday night, Saturday night, gives you 1.5 days. So Sunday noon, how about Sunday 12 noon? All right, that work? Okay, so to recap, the Monday lecture will be due Wednesday midnight, and the Friday lecture summary will be due Sunday 12 noon. All right. Okay. Lisa, you're good?
Yeah, I'm good.
All right, great, thank you. Super. So now we just need to pinpoint what time.
Those would be the best days as well, the Monday and the Friday. Very late.
Is it?
Or for you, I guess.
Yeah, for me, yes. For me, yes, because then at least the middle of the week, I have the full day, right? And then just Monday and Friday, we have this. So how about, okay, so enough time for having lunch and all that. How about Friday, 2 o'clock? I mean, yeah, Monday and Friday, 2 p.m.,
Oh, okay.
2 to 3:30? Yes? Okay. All right, so Monday, Friday, 2 to 3:30. Super, thank you very much. All right, that also allows me to do this Friday lecture because it happens that I'm going on retreat, my yearly canonical retreat as a priest today from this afternoon to Friday morning. So I can do the lecture. I may have to cut it short a little bit, that one lecture Friday, because I have another meeting at 3 o'clock, but I'll work on that. Okay. Oops. Water. Yeah. There. All right. Yeah, gotta leave it alone. All right, any other technicalities? I'm sure other technicalities will come up, or we'll address them as they show up, okay? Let's get right on to it. So we are shooting for 8 weeks, which will mean 16 classes. And we have modules, and I have some posting there, weeks 1 through 2, etc. on Canvas. You can follow that. You have access to Canvas also. All right. And let's see. So the first 2 weeks, I want to focus on the bioethical issues at the beginning of human life. The next 2 weeks, I want to focus on bioethical issues at the end of human life. And then the last 4 weeks on bioethical issues in healthcare in the middle of life somewhere, okay? And some issues with beginning of life, I'm putting into healthcare because we just don't have enough time. For example, ectopic pregnancies and so forth, looking at it from the perspective of the mother, will be healthcare, and then we can address them then, all right? All right, so beginning of life. Also, I want to say that this whole course is being taught from the principal perspective of bioethics. So let's talk a little bit about that. It's conceptual, but it's also existential, okay? It's important because we have certain frames of reasoning that we may or may not be aware of. Many times it's subconscious, but they're frames of reasoning anyway. And a lot has to do how we come out, how we analyze a bioethical issue on our frames of reference. And I can tell you that it boils down to 2 basic ways of looking at ethical issues, because sooner or later, we're passing judgment on human behavior, right? Whether some things are right or wrong, that's ethics. We never pass judgment on the conscience of people, by the way, at least from the Catholic perspective. We don't pass judgment on the conscience of people. That's subjective, all right? But we can judge behavior. So let's talk a little bit about that. How do we judge behavior without judging the person as such? Well, because actions in themselves may be right or wrong. Whether the person is culpable or not is a different story. That's a conscience issue that belongs to God, who has access to our conscience. But we can say objectively some things are right or wrong, right? For example, I hope that you agree that killing an innocent person is wrong. And in fact, that's even codified in the law. It's called murder. So murder is wrong, right? Always and everywhere, because it's not on the situation. What would be an exception where we could justify conceivably the killing of another human being? Self-defense, obviously. If someone comes at me with a knife and I happen to have a gun, then it's bad luck for the person with a knife, right? And so we have a right to self-defense. It's a natural right, which is also protected by civil law. But it's a natural right that we have because we're alive. We have a right to continue to be alive, and we have a right to self-defense. That's at the individual level. We can also expand that right to at the national level, for example, which is a right to have a military, again, for defense. So murder implies that the person that is being killed is not innocent but guilty, or rather not guilty but innocent. That's the murder, okay? And whether the person, the person doing the killing is culpable or not in their conscience, that's a different story. But objectively, the act itself, the behavior of killing an innocent human being is wrong. It's objectively wrong. And that's how we can do principled bioethics. Now, the alternative to principled bioethics is what is called utilitarianism. Let me go back here. Just, open. Let me see. I want to put in this slide. So it's actually easier to do it on Word. This is a whiteboard. So bioethics, cellular principle, or utilitarian. All right. And at the end of the day, it's going to be about the means and the ends. So we got means and ends, right? And the end may be a good end, right? But the means in principle bioethics also have to be justified. The means also have to be good, all right? So we say something like in principle bioethics, the end does not justify the means. The means have to be justified in themselves. Whereas in utilitarian bioethics, the means are justified by the ends. Whether they want to admit it or not, that's the bottom line. I've been in this field for decades, and that is my bottom line conclusion, right? So to give you a simple example, let's say I'm doing an act of charity. So the act of charity in itself is good. The end is to give something of my own belongings, of my own property or money to someone else who is in need. So that in itself is a good thing, right? That's the end. But the reason for doing it could contaminate the act itself. For example, I may do it for the charity itself out of generosity, or I may do it just to lower my tax bracket, or I may do it because I want people to watch me and feel, get the praise of someone else, trying to get the attention of somebody else, etc. So it all depends on how I'm doing, why I'm doing that action. The why, all right, is the means. And so the means have to be good in themselves also. And that's very critical in bioethics because we run into techniques and technologies that allow for things to be done, but those means also need to be justified. And I can tell you that right here, right now, we part waters with utilitarianism, with utilitarian bioethics. Because for them, essentially, the end is a good, noble end, positive end, and therefore whatever means to get there, as long as it's legal, of course. So they're not asking us to break the law. All right, as long as it's legal, then the end will justify the means. And the means typically then will be driven by technology. If it can be done, then it should be done automatically because we can do it technologically. You see, there's a danger, there's a challenge there, right? And so what they say is, well, if you think this is so bad, then make it illegal. But while it's legal, please step aside because we can do it. That's the challenge forever with utilitarianism, is to enter into really substantial dialogue with these people that some things should not be done out of principle, And that's why it's called principled bioethics, in contrast to utilitarian. Under utilitarian ethics, it's also other aspects of can be further refined into legalism, for example, as long as it's legal, right? So if you think it's so bad, then make it illegal. In other words, go through the legislative process. The challenge with that is that not every aspect of our human behavior can be codified into law, because then that law will be extremely oppressive. I'll give an example. I think that if we're honest with ourselves, and of course, principled bioethics, together with utilitarian, implies that we're talking to normal, rational human beings, all right? There's always going to be the exception of someone who's fanaticized or radicalized, or someone who has invincible ignorance that no matter how much argument we present, they will not be convinced because they have some kind of block that is impeding them from seeing the truth, all right? But I'm talking about for normal, rational human beings, the vast majority of us, the argument of reasoning. And so, for example, I think that any normal human being would consider adultery wrong, okay? And if someone has a question about, well, what if it were happening to you? What if your spouse or your boyfriend or girlfriend, is cheating on you? How would you feel about that? And then I feel good. You're going to say, oh, keep doing it. It feels great. That's not normal, okay? So we assume in an intimate relationship like that, that there's going to be exclusivity, that there's going to be fidelity, right, to each other. And so adultery, normal human beings can agree that should be wrong. It's wrong. It's not proper behavior. It's not good behavior. But it's not codified. I mean, when was the last time you saw someone getting arrested for adultery? It's happening right now, and they don't get arrested, right? In fact, it's in some books. It used to be illegal, and in some states, it's actually harder to get some laws off the books, more expensive and cumbersome. So they just leave them there, but they don't enforce it. All right. So even in a few states, you could still find that there is a code, there is a codification of adultery as illegal, but people don't get arrested because it's not enforceable. Are you going to go into people's bedrooms or what?
Right.
Or have cameras? So it's just too much. It would be too oppressive. But we have to assume that normal behavior will develop, people will develop a code of ethics that allows them to live in harmony with the rest of society in a civilized way without having to be told by law how we have to behave all the time. All right? And so that's the argument that I try to make for principled bioethics, that it's impossible to codify everything. That law, that type of regime would be really a totalitarian regime. Something that we would expect out of communism or a dictatorship or a tyranny, where they try to manage every aspect of a person's life, even cameras that monitor their movement, the restrictions of moving from one place to another. I know the pandemic was an extraordinary time, right? But under normal situations, we have freedom to move around and so forth. Okay, so this whole course is taught by the assumption of principled bioethics, which are values that we can uphold always and everywhere. So if murder is wrong for us here, it's also wrong for someone in Tanzania or Greenland, any other part of the world, okay? Because human beings, we have a common humanity, we share common humanity. As we go along, these principles will be showing up in our analysis. So this is the one that I want to talk about for now, this principle bioethics. Okay, so this is a little bit of preamble. I'm going to get into the beginning of life, because what happens here is the question of when human life begins is a crucial question, right? Depending on when human life begins, how our bioethical analysis is going to be. The issues like abortion, contraception, in vitro fertilization, natural family planning, which are the 4 big topics that we're going to cover this first couple of weeks, all right? So when human life begins, and I'm talking about organic life, right? Again, not the life of angels or something like that, that is metaphysical, but a physical, empirical, organic life as we know it here on Earth. We know that organically we are mammals, and so we belong to the animal kingdom with all the characteristics, all the benefits and burdens of other mammals and animals here on Earth, meaning that we need to eat, sleep, etc., and metabolize. So we can determine when life begins, because the beginning of human life is no different from the beginning of any other life, or any other individual life. Particularly mammals, we're going to focus on mammals so far simply because the fertilization is fairly well established. We know that we have beyond internal fertilization, we have also internal gestation, right? So there's a pregnancy period time that's standard for mammals. Yes.
I know, because I read ahead, like I know where you're going to be going with this, but
Yep.
I wanted to know, like, so a cell is considered alive. Of course, right?
Okay, so is a cell alive?
What kind of distinction?
A cell is either dead or alive, right? And the distinction between the two is, again, thinking organically, right? What's going to be a distinction between a living cell and a dead cell? We actually have both. Right now, if we look at our skin, the outer layer of skin is dead, right? You know that we have layers of dead skin. It's a good thing, actually. It protects us. It protects the living cells underneath, the epidermis. But below the dead skin, there's going to be live skin, epidermal cells, right? And those are live. So what's the difference between those two layers? We can take it down to the cellular level. One is metabolizing and the other one is not. So organization, order. Metabolism, which you know is anabolism and catabolism, you're familiar with all that, right? Building up and breaking down of organic molecules, but in an orderly fashion, cascades, etc. And so if that is happening, there is order. There is a metabolic internal order to that cell, and therefore that cell is alive. It's metabolizing. When metabolism breaks down, apoptosis or whatever is happening to that cell, then that cell is dying or dead. The metabolism is actually breaking down. There's disorder introduced. Okay, so yes, a living cell is alive.
Well, no, but I meant, sorry, I meant like, so it's, for example, egg and sperm, but then embryo.
We're going to get there.
Okay.
We're going to get there. We're going to get there rather quickly, again, because of your biological background. Here is the PowerPoint that you have all access to. And those who wish, well, you can review the, you can review. So I'm going to focus on sexual reproduction. There are two modes of reproduction in nature that we know of, right? Sexual and asexual, non-sexual. I'm not going to talk about asexual. There's a number of different varieties of asexual reproduction, but we're focusing here on sexual reproduction, which is the characteristic for mammals. There's a little review of mitosis there that you can look at, no problem, you know. So just to throw out, you know, replication, which is what? Replication is chromosomal duplication, right? It happens in the synthesis part of the cell cycle just before mitosis, so that the two daughter cells end up with what? The same number of chromosomes. So you're familiar with diploid and haploid, all right? So let's just start with a single cell that is diploid and undergoing mitosis. For the, how many cells are going to come out of a normal mitosis? How many cells? Two daughter cells, right? From one mother cell, two daughter cells. Those two daughter cells, are they going to be haploid or diploid in mitosis? Diploid. But the mother cell is also diploid. So what needs to happen, just arithmetically, for the two daughter cells to be diploid, to have the same number of genes, right? What needs to, or chromosomes, what needs to happen to the mother cell before the split? Replication. The number of the 2n needs to duplicate into 4n. So there's a temporary 4n stage of every cell before undergoing mitosis, right? So that then that 4n gets split up into two 2n's. So simple arithmetic. That simple arithmetic, you'd be amazed how many people get confused right there. And they trip and stumble and they can't figure it out. But keep in mind, replication is the same as duplication. Keep in mind that in science, we don't want people to understand what we're saying. So we use these fancy words, replication, but it's really duplication, right? It's duplication of chromosomes. And that has its own machinery and so forth. Okay. So moving forward then, really what I want to get into is meiosis, which I think is, it's a good one. Let's start here. Okay, so meiosis is like it's a double mitosis, but with a twist. So just to review, most cells in our body undergo mitosis all the time, right? For cell growth and tissue growth and so forth. But a few cells in our body undergo meiosis, and because the object of meiosis is to produce what? The sex cells or the gametes, all right? Gametes. And so gametes, are they diploid or haploid? Haploid. right? They don't have a pair, they don't have chromosome pairs, they're haploid. And so that is known as, have you ever heard chromatic reduction? The word, the little phrase, chromatic reduction. Chromatic reduction, so this is a language that you need to be familiar with, okay? Just on the science side. That's what meiosis does. It goes from 2n to n. So it's just like, this is like an algebraic formula that the coefficient when it's 1 is not used, it's implied, it's not shown. So this n, what is the coefficient of that n? 1, all right? Okay. And n stands for number of chromosomes, number of chromosomes. Straightforward, very simple, don't get confused. So chromatic reduction is meiosis. It reduces the number of chromosomes to the haploid number, from diploid to haploid. That's called chromatic reduction. All right. So we're in meiosis here. And meiosis, like I said, it's a double mitosis, but with a twist. You know that in normal mitosis, when the chromosomes line up at the equator, then they split, right, to opposite poles, but they split singly. So the pairs split up at that point, and you get one set going to one pole, the North Pole, and the other set of chromosomes going to the South Pole. If we're talking about the human, how many chromosomes total are we talking about? Total, diploid number. 46, or in pairs, 23 pairs. Okay, so 23 pairs, 46 is the same thing, arithmetically the same thing. Stay with the numbers for now. All right, so 46 or 23 pairs. 23 pairs implies a diploid cell. 46, the total number. So at mitosis, at the prophase, going to metaphase, they split up. 23 go to one pole, the other 23 chromosomes go to the other pole, and then eventually you get cytokinesis, right? And 2 daughter cells. Right? Now, in meiosis 1, what happens is, so meiosis can be divided into meiosis 1 and meiosis 2. It's a double split. In the first split, those chromosomes migrate to the opposite poles in pairs, not singly, but in pairs. So that at the end of meiosis 1, in each pole, you end up with 23 pairs. Okay? And then meiosis 2, for each one of those 2 daughter cells, meiosis 2 is a simple mitosis. At that point, those chromosome pairs do split up. See, they split up to get the haploid number. So the net result in meiosis is from each mother cell, how many daughter cells? 4. 4 instead of 2 because it's a double split, right? But because of the splitting of the chromosome sets in meiosis 2, then the 4 cells end up diploid or haploid? Haploid. You see, haploid. So there's still one replication going on, but that one replication now has to be redistributed into 4 cells instead of 2 cells. And that's why we end up with haploid number. All right. So again, it's simple arithmetic. Just keep in mind that meiosis involves a double split with the little twist that in the first split, the chromosomes stay in pairs. And then in the second split, it's a simple, it's basically straight up mitosis. Now, why, again, there's a purpose. Why do and these are the gametes, right? They're haploid. We also need to know that they're complementary. So there's male gametes and female gametes. And why is that chromatic reduction? Because what is the function of gametes? The function of gametes? Fertilization. To reunite, right? To reunite. And so it's really a unique process. When you think about fertilization histologically, it's the only case that I can think of where you have 2 cells merging into one and the result is viable, okay? And it's a good, happy result. In other words, it preserves the species, it continues the species of whatever species where fertilization is happening. Because normally, the normal scenario for cells is not to fuse, but to split, either mitosis or meiosis, right? That's the normal function of the normal process of cells over time is to split and keep splitting. But here in fertilization, we have this unique process of two cells coming and fusing into one and surviving and thriving the process. There's one other case that I can think of when two cells are fused into one, but it's not a happy ending for one of the two cells. Can you think of that case? Yes, phagocytosis. Remember when, say, a white blood cell engulfs a bacterium? It's not a happy ending for the bacterium. It's going to get digested up. All right, the phagocyte survives. So that's the only other case that I can think of where two cells actually come together, but not a happy ending for one of them, for sure. But in fertilization, yes. Now imagine if there was no chromatic reduction, if the gametes were diploid. If they fuse, if you get a diploid egg, hypothetically with a diploid sperm fusing, right? What do you get? 4N, the tetraploid. You get a 4N cell. Right? You get a 4N zygote, which is that viable? Not viable because that codes for double of everything. 2 heads, 4 arms, 4 legs, or 2 hearts, double of everything, which is certainly not viable. You follow? And so that chromatic reduction has come through evolution for establishing this elegant process. Another way of, well, many ways of describing, but this elegant process of fertilization to pass on the genetic material down to 2 little microscopic cells, 2 gametes that are complementary, so they fuse and they form a new cell called the zygote, which is a new individual of any species, particularly, well, we stay with those that reproduce sexually, of course, which, by the way, is not just plants, it's not just animals. You know that plants also reproduce sexually, right? What is the equivalent of sperm in the plants? A yellow dust. You just washed your car and now it's covered with this yellow dust.
Yeah,
pollen grains by the millions, right? In fact, each pollen grain contains 2 sperm nuclei inside normally, all right? One sperm nucleus does a little hole through the stigma, and then the other sperm nucleus travels down and fertilizes. The egg, which is going to be at the base of, if you think of a flower, the pistil, is where the eggs are. So even in plants undergo fertilization, and you can look up any standard textbook will show the first cell product of fertilization is called the zygote. I want to get to that one. Any questions or comments or clarifications on this thing? No? Okay, we know that gametogenesis then can be subdivided. Oh yes, the reason for this one is there is already a difference here in the sexes, in the male and the female, because the top one represents the formation of, so this is spermatogenesis, the formation of sperm. We end up with 4 sperm from every primary spermatocyte. Okay, we end up with 4, let's say, functional sperm haploid. But in oogenesis of the primary oocyte, we end up only with 1 functional egg and the other 3 polar bodies. There have to be 4 cells, right, for the haploidy thing to work, right, for the other cells. But what happened to the other 3? They're called polar bodies. And maybe this is something that people don't know, because we know that normally on ovulation, there's only 1 egg produced on average, but the other 3, it's because in meiosis, the split in meiosis 1 and meiosis 2, the split is uneven. The cytoplasmic split is uneven. So for the first split of meiosis, one of the 2 taller cells ends up with the bulk of the cytoplasm. And the other daughter cell ends up with very little cytoplasm. It's non-viable. Not viable to sustain a fertilization. So that's this unevenness doesn't occur in spermatogenesis, but it does occur in oogenesis. So obviously, somehow the female genes are driving that uneven cytoplasmic cytokinesis. All right, and that's meiosis 1. And then when they split again, the daughter cell that is degenerate, that has less cytoplasm, will again split with even less cytoplasm, distributing the little cytoplasm that it had into 2 daughter cells. But the other one also, there will be an uneven split again in the second meiosis on cytokinesis, right? So that the end result is that of the 4 daughter cells, 1 of the 4 ends up with the bulk of the cytoplasm, and the other 3 end up just with the same amount of chromosomes, with haploid number of chromosomes, but very little cytoplasm. And they're so small that they actually hang around and stick. Also, we know that the egg normally is not just by itself, in contrast with the sperm, which are individuals, but the egg is actually a cumulus. It's a whole cluster. It has a corona radiata and a zona pellucida, has 2 layers. Each egg, each ovum has 2 layers of cells that is protecting the egg. And embedded in between those layers are the other polar bodies. I'll actually show you some photographs, micrographs of that coming up. So basically, we know that functionally this occurs in the sex organs and the ovaries or testes, right? And so that's the diagram there. For the testicles, we end up with 4 sperm cells for every primary spermatocyte. But for the ovary, for each follicle, each primary oocyte, we end up only with 1 ovum, functional, and then 3 polar bodies that are hanging around the layer there. So we can go here, for example. Well, these are just to scale, you know this. All right, I want to get to the bottom line, which is syngamy. Syngamy is the completion of the fertilization process. All right, so we can talk about, we can say philosophically, the moment of conception, but organically, we talk about the process of fertilization, which takes up to 24 hours for the fusion to occur, from the beginning of the penetration of the sperm through the corona radiata outside here, and then the zona pellucida, and eventually, so the sperm at the tip has a little gland called the acrosome, maintaining digestive enzymes. So when that acrosome makes contact with the zona, it spills out, with the corona, sorry, it spills out the digestive enzymes, and it literally bores itself through those 2 layers until it gets to the membrane of the egg, and then literally pours its nucleus inside the cell, inside the ovum. All right, and those nuclei, just to this, because they're haploid nuclei, they're called pronuclei. So you run across that word sometimes, pro-nucleus, are haploid nuclei of the sperm or the egg. So the 2 pronuclei need to fuse together to form the new diploid nucleus of the zygote. Okay, and so we say that fertilization restores what? Functionally,
The number of chromosomes.
yeah, well, the diploid number.
Yeah.
Diploidy. That's why it's essential that those 2 gametes be haploid, okay, the complement. So fertilization restores diploidy. These little sentences or phrases are packed with meaning, all right? It's describing what fertilization does functionally. Structurally, all of the background that needs to happen for that to occur. You think of structurally, beginning with functional, mature sex organs and so forth to produce the whole process, and then the actual mating process, everything else. But it's standard. In other words, this is the same process that occurs essentially in any mammal that is reproducing, okay? They have to get eventually to syngamy. In the human, as in other mammals, here's just a diagram of the female reproductive tract, mostly the fallopian tube. It occurs in this area, the distal end of the fallopian tube called the ampulla. It's also known as the horn or the bend of the fallopian tube. Of course, this is not to scale. If the zygote were that huge, it would be too large, all right? We're talking about a microscopic cell, actually. But for the purpose of diagram, it is illustrated that large. This little area, which is called the infundibulum, is like a little glove that is next to the ovary, not attached, but close to the ovary. And actually, when this infundibulum comes close to the ovary on ovulation, is to catch that egg, that ovum, when it ruptures, and then the ovum will sit here for about 24 hours on average, waiting for a sperm to be fertilized. Okay, now we'll look at a little bit more. Then we'll get into natural family planning, some details about that. But basically, it just takes, how many sperm does it take to fertilize one egg? One sperm, 1 to 1, all right? And yet, for the human ejaculate to be considered clinically potent, for man to have clinically potent ejaculate, you know how many sperm it needs to have? About 100 million sperm. 100 to 150 million sperm is considered a potent ejaculate, capable of fertilizing. So the vast majority get lost on the way, right? So we can see that the selective pressure against sperm is about 100 million to 1. Just unbelievable. That's what we can say on average. It is the strongest, most vigorous, most capacitated sperm that gets to fertilize the egg, on average. You see? And that's why, on average, the ones who are born nine months later are the most vigorous possible humans from that fertilization. Because the weaker sperm got left behind.
So natural selection acts on both stages, haploid and diploid.
Yes, there's natural selection all the way, but this one is incredibly powerful. The other stage of that, there's 100 million to 1, okay? Super strong. This is actually one of Darwin's 5 observations for the theory of evolution, the Hampton theory of evolution, which is a pre-zygotic overabundance of gametes. There's like a waste, all right? There's pre-zygotic and there's post-zygotic. In other animals, many eggs are actually fertilized, like in fish and so forth, but the vast majority of them don't get to survive, they get eaten up. So that's post-zygotic selective pressure, all right? It's all in the big picture of natural selection. So really, again, we say it's elegant, right? Can we go to 12:15 today?
Yeah.
Yes, all right, appreciate it. All right, so you see what we have is the other selective process is going to be after fertilization. If fertilization does happen, then this little zygote has to move down into the endometrium of the uterus, which is the proper place for implantation, all right? Which is, it takes, for the human, it takes an average of 5 to 7 days to make that voyage. 5 to 7 days, about a week, for implantation to happen. Okay, and during those 5 to 7 days, that zygote is already developing, is undergoing what? Mitosis. After fertilization, it's mitosis forever. Mitosis, your tosis, everybody's tosis. It's just mitosis all the way until the grave, all right? And so it's going to go through the 2-cell stage, 4-cell stage, 8-cell. It's a geometric progression, right? It's going to go through the morula, blastocyst, and it is the blastocyst that implants. It's actually the 3rd stage of embryonic development. It's the blastocyst that implants. There are biological reasons for that. But that's the other selective process that it's estimated that about 50% of human pregnancies never make it to term naturally. I'm talking about miscarriage or spontaneous abortion. I'm not talking about a procured abortion. Natural losses, right? About half, 50%, depending on who you read. It's actually a fuzzy number. It's difficult to track because many of the losses occur in the first couple of weeks of pregnancy. The blastocyst doesn't implant for whatever reason. Typically, the vast majority of those that have been observed and checked, they carry chromosomal abnormalities. Okay, so they're non-viable, and the endometrium naturally discards them. It's a natural loss, and it's the other selection. So we can say that post-zygotic and pre-implantation, there's another 50%, there's another 2-to-1 selective pressure against the embryo. But the biggest by far is on the sperm, pre-zygotic, okay? Right. Everybody peachy keen with the terminology and so forth and the concepts? Here are a few micrographs that you can see. Large enough, yeah. Okay, so here we have a electron micrograph of a zygote. You can still see the 2 pronuclei, the male and female. We don't know which one is which, but those are the 2 pronuclei there. And you can also see a couple of the polar bodies. You see up here? Those little blobs,
yeah.
the 2 little blobs, those are polar bodies that are stuck in between the zona pellucida and the corona radiata. Those other 2 layers of cells that protect the egg, all right? So it's a neat photograph that shows everything in place. Eventually, when these 2 pronuclei fuse finally to restore diploidy, that's the completion of syngamy, that process called syngamy. Then it goes into the moral stage, 2-cell stage, 4-cell, 8-cell. Eventually, you have a cluster of cells. Still, each one of these individual cells is called blastomere, which is, they're all totipotent. You're familiar with potency? Totipotent, pluripotent, fully multipotent, and then fully differentiated, right? The 4 stages. So we're all hunky-dory on that. All of these are totipotent. The zygote is also totipotent, all right? But then what happens when you have a cluster of cells, the first cellular differentiation happens. First cellular differentiation, which is that ball of cells begins to hollow out on the inside. Hollow out, all right? Think of a tennis ball or a basketball. Now there's no air in there because there's no air to be had. It's fluid, mostly water, fluid, all right? But it hollows out. So that what happens is you start getting 2 different cell layers. There's going to be an outer cell layer that is normally known as the trophoblast, right? And then there's an inner cell layer here, an inner mass of cells that is called the inner cell mass, unimaginatively, or the ICM, the inner cell mass, right? Sometimes you also see it as the embryoblast, right there. So trophoblast and embryoblast, 2 cell layers is the first moment of differentiation. At this point, the blastocyst then is no longer totipotent, it's pluripotent, all right? In fact, it is now known that the embryo proper comes from the embryoblast, from the ICM. The embryoblast becomes the inner cell mass becomes the embryo proper, going to undergo gastrulation and all that. And the trophoblast, what does the trophoblast become? If you think about it functionally, again, what is embedding on the endometrium? This is the endometrium, the inner lining of the uterus that has become spongy for accepting a pregnancy. It's the trophoblast that is in direct contact with the endometrium, right? So it stands to reason the trophoblast is what becomes the placenta. You follow? With vascularization. And the trophoblast has a very unique challenge to convince the endometrium that he or she is not a parasite, not foreign, but is a friend and not a foe. So the pregnancy is not rejected for a normal pregnancy, okay? Because what is the endometrium saying about this trophoblast, about this blastocyst that is embedding into it? Ambiguous signal, right? 50% of the genome comes from the mom. So 50% of the genome, I'm talking about the blastocyst, 50% of the genome of the blastocyst is identical to the mother because it came from the egg. But the other 50% came from the sperm, who is another individual, the same species, but a different individual. So it's foreign to the mother and to the endometrium. And so the endometrium is ambiguous about this thing that it's implanting into it and demanding, challenging to establish a network of capillarity, all right? And so that's why 50% of the pregnancies approximately get to survive the other 50 don't, because that match, again, has to be down to the biochemical level. All right, and if there's something wrong, then the endometrium says, no, go away.
So the selection is against basically trophoblast genes.
Okay, the short answer, I don't know. I'm not an embryologist to get into the
that's
detail,
fine.
but you can take it to that level, right? Keep in mind that gene expression is typically a very complex thing, has to do with the environment also, and alternative folding and so forth. So it's very, very complex, but we can say for sure that it's down to the biochemical level.
Right.
All right, and everything has to be in sync, all the cascades match up so that the endometrium actually allows that trophoblast to insert and to establish it's called the syncytial trophoblast. It continues with different stages of establishing the placenta. Once the placenta is established, we know that there's a lot of back and forth going on now, not just chemicals. Of course, it's a dumping station for the embryo, right? Through the placenta, has to get rid of especially the gas exchange, but also even actual cells go through. Maternal cells go into the fetus and fetal cells go into the mother. So we end up with what is called the microchimeras. Moms that have had pregnancies, they have areas in their body where they have some fetal cells. And fetuses, we all carry some mother cells in us. So they're called microchimeras. It's very interesting. Yes, we are in a sense. But it's still the same species because at worst is our father's genes, right, that are kind of foreign to the mother. All right, other stages of development, again, moving forward. This is the famous Carnegie stations of embryonic development. This is kind of the standard in embryology, the Carnegie stages. And what I just want to point out by this is that philosophically, there is no quantum leap between one stage of development and the other throughout the 9 months. From the zygote to birth, what we have basically is growth and development of cells, differentiation into tissues into organs into systems, and so forth. In fact, it is now known that in the first 8 weeks approximately, all of the organ systems are already formed and in place in the embryo, but miniature because the little embryo is about 1 inch long, more or less. So they're all in place in miniature, and from there on, it's mostly growth in size of the organs and tissues, and so it also changes name from embryo to fetus. Fetus meaning a tiny being in Latin, all right? But it's a technical change in name. There is a progression between the stages so that at no particular stage, we go, let's say, from non-human to human, from another species to the human species, or even from unspecific life. That's another point. I'm interacting here. You have to follow because the arguments go back and forth between biology and philosophy, all right, or theology. But there's no substantial change, meaning no change in species in any stage in the embryology for any species, not just a human. You can think about an orangutan, a kangaroo, or any other mammal. But there's also, so there's no leap from one species to another, and there's also no leap from non-species to species. In other words, I can ask the question, can you think of any animal, any organism that is non-specific, that doesn't belong to a species? There's no undifferentiated organism. Every organism, by definition, belongs to a particular species, has a genome, all right? And so that's a beautiful, again, elegant. There's an elegant matchup between biology and philosophy at the level of species, what we would call a substantial being. So hippos are substantially hippos philosophically, and organically they're hippos because they have a hippo genome. The same for human. Because we have a human genome, we are substantially human, all right? And we belong to Homo sapiens. And so there's a matchup between the biological datum and the philosophical analysis that we are all substantially human. And we're substantially human at every stage of development, forget about the unborn for now. Let's think of the born, right? A baby is obviously less developed than a teenager, and a teenager less developed than a human. Is a baby less human? That's actually an offense, you know. It's disrespectful to say that a baby is less human simply because he or she is less developed. The same with a toddler or a teenager, right? We're human through and through. We're in stages of development, for sure. That's obvious that there are stages of development. But humanity as such does not afford gradation, what we would call gradation, right? This is another one of those little phrases or sentences that is meaningful. Right? Because, and sometimes here's a little tool. Here's a little tool that we can use, again, in bioethics and in philosophy in general. When a statement sounds a little strong, flip it and see if the opposite makes sense. Imagine if humanity did afford gradation. In other words, we would be able to say that some humans are more humans than others, or some humans are less humans than others. That actually has happened historically, right? It led to what? It led to enslavement. It led to selling human beings on the market and branding them as cattle or things, right?
And also things like the Holocaust.
And the Holocaust, thinking it definitely, Hitler thought that the Jews were the scum of the earth. They were cancer, and they had to be eliminated. It's insane to think in those terms today, right? So we have to uphold the humanity of all human beings. But the point is that we cannot start with birth, because by birth we have been living for 9 months already, and we have to deal with the human embryo and the human fetus, you know, as human. Okay, so you see where I'm going with this argument, that essentially granting not granting, because we don't grant anything, recognizing the humanity of the human embryo. And then the next step is that because they are in an other stage of development and dependency, they need advocacy, because they cannot speak on their own. They need advocacy, right? And that's, again, where the ethics argument comes in. Okay, run out of time. So we're going to cover abortion on Friday. But you can see that I've set the stage here. Pretty much, if you've been following the argument, analyzing the issue, it should be fairly straightforward, even though it is one of the most loaded and controversial issues of our time for decades already. Okay, but we do a bioethics also that is based on what we call natural law. In other words, we allow nature to speak to us and tell us at least the objective facts, and then we do the analysis on that. Let me stop and ask if there are any questions or comments. All right, well, you can think of some questions if you want for next time. The slides are there, and we'll try to do abortion and contraception Friday, and then in vitro fertilization, natural family planning next week.
So
Yes.
this lecture summary is due Wednesday.
So this is a lecture that will be due Wednesday by midnight. All right.
We have class Friday at 2.
Friday at 2, starting this Friday, 2 p.m. Yes. Thanks for reminding me. So this goes to 2 p.m. Yes. So thank you very much. All right.
Yes, thank you so
Looking
much.
forward to continuing with you. So let me close the recording now.
Would anyone like dry Russian fish?
That's
Mhm.
good thing I have the mask on here.
Does it
Yes.
smell like fish?
We can go fishing maybe.
Subtly. Yeah.
Use it as bait.
It looks like bait, yeah.
No, but it's